Hello everyone and welcome to today's session, and today we'll be discussing the theory related to chapter Human Health and Diseases. This is the part 2 of the lecture, and in today's session we'll be discussing NCERT section 7.2. This is the disclaimer. So what are the objectives of today's session? So today we will understand the basic concepts of innate and adaptive immunity. We'll also talk about their components and mechanism of its action in protecting human body. Then we'll differentiate between active and passive immunity. Okay, so there are two types of immunity, active and passive immunity. We will differentiate between these kind of immunities and describe the role of antibodies and antigens in immune responses. We'll also understand the process of vaccination and immunization, including the importance in disease prevention. So these vaccines, they are very important. These vaccines consist of attenuated pathogens, okay, which is weakened pathogens. These weakened pathogens, they are introduced into the body, and immune response generates in response to these weakened pathogens. Okay, clear? So we'll talk about the vaccination and immunization as well. So today we'll be talking about the topic immunity, which is NCERT section 7.2. Then we'll summarize the entire section. We'll also discuss some NCERT-based questions. We'll discuss some keynotes that will help you memorize these topics, and in the end, there will be some interesting questions that will help you enhance your critical thinking ability. So let's start with today's session. So let's talk about the definition of immune system or immunity. Okay, so let's understand what immunity means in the context of human health and diseases. So immunity is the overall ability of our body or the host to fight off disease-causing organisms which are known as pathogens. Now, what are the types of immunity? So there are two types of immunity, innate and acquired immunity. So our bodies have two main ways to protect themselves from harmful invaders like bacteria and viruses. So these are innate immunity and acquired immunity. So you can think of them as different defense systems working together. Now, innate immunity is body's immediate rapid response to an invasion, and this happens within minutes or hours. On the other hand, acquired immunity is delayed compared to innate immunity, taking days to weeks to fully activate upon the first exposure to the pathogen. Now let's talk about the specificity. Innate immunity is non-specific, meaning it attacks any foreign invader it encounters, not targeting a particular type. But the acquired immunity is highly specific, meaning it recognizes and targets particular pathogens. Now let's talk about the memory. So innate immunity does not have immunological memory, so it reacts the same way every time it encounters a pathogen, whereas acquired immunity have immunological memory. Once it encounters a pathogen, it remembers it, allowing for a much faster and stronger response if the same pathogen is encountered again in the future. What are the components of these two immunities? Let's talk about the innate immunity first. So innate immunity includes the body's initial defense like physical barriers, which include skin, mucous membrane, as well as phagocytes like macrophages, and the process or inflammation. This is also included under innate immunity. On the other hand, acquired immunity involves specialized cells called B cells and T cells and the antibodies they produce. Now, where are these kind of immunity present, or at what time they are present? So, innate immunity. So, it is present from the birth. Okay, so the animals, they are born with innate immunity. Now, acquired immunity develops after the birth as you are exposed to different pathogens, or it develops through vaccination. Okay, so let's talk about the type of immunity. We were talking about the type of immunity. You're talking about the innate and acquired immunity. So, Let's first talk about the innate immunity. So innate immunity, this is evolutionary older and more primitive system and mounts the same non-specific response to a pathogen every single time, and it provides short-term protection from the birth. In contrast, acquired immunity, also known as adaptive immunity, is a more recent evolutionary development that provides targeted long-term protection. Now, defining feature of acquired immunity is immunological memory, which allows the body to remember a pathogen after the initial exposure, leading to a much stronger and faster response upon each subsequent encounter. Now, these two systems work together with innate immunity, providing immediate defense, while acquired immunity develops a more specialized and lasting one. Okay, now let's talk about the innate immunity in detail. So, we have already discussed that innate immunity is like the body's first line of defense. Okay, now innate immunity includes physiological barriers. Okay, let's talk about these two: physical and physical barriers and physiological barriers. So physical barriers are like skin provide a tough outer layer, while the sticky mucus coating in our respiratory, gastrointestinal and urinary tract help traps the microorganism. What is included under physiological barriers? So physiological barriers include acidic stomach, juices that kill most ingested microorganisms, and protective fluids like tears and saliva, which contain antibacterial enzymes such as lysozyme. Okay, so here you can see these physiological barriers include the stomach juices, digestive juices, as well as the fluids like tears and saliva. Now, together, these barriers create a hostile environment that stop pathogens before they can cause infection. Okay, now innate immunity also includes cellular barriers and cytokine barriers. So beyond the physical and physiological defense like skin and stomach acid, innate immunity system has two other critical barriers: a cellular barrier composed of specialized white blood cells and a cytokine barrier that use signaling proteins. Now the cellular barrier involves various white blood cells or leukocytes including PMNL, neutrophils, monocytes, okay, and natural killer cells that circulate in the blood and tissues to actively phagocytose invading microorganism. The cytokine barrier is made up of proteins called interferons, which are secreted by virus infected cells to protect healthy non-infected cells nearby, interfering with the viral replication. Together, these cellular and chemical components provide a robust, non-specific, and rapid response defense against the pathogens. Okay, so you should know what all is included under cellular barriers and cytokine barriers. So, cellular barriers means the leukocytes, also known as WBCs. Then the type of WBCs are also mentioned, and then cytokine barriers basically include chemicals like interferons. Let's talk about the acquired immunity now. Now, acquired immunity is pathogen specific and characterized by the memory. This means our immune system can recognize and remember specific pathogens, making future responses faster and stronger. Okay, now let's talk about the primary and the secondary immune response. Here you can see in this table, okay, the characteristic of primary immune response and secondary immune response. Now, the primary immune response occurs upon the first encounter with an antigen or a pathogen and is typically feeble to moderate in intensity. Okay. Now, as the immune system must identify the invader and begin creating specific antibodies and memory cells. Okay, clear? So this is what is primary immune response. It gets activated upon first contact with the pathogen, and its intensity is feeble to moderate. Clear? Now, let's talk about the secondary immune response. Now, secondary immune response happens during subsequent exposure to that same pathogen. Okay, clear? So, thanks to the memory cells created during the initial response. So, this reaction is significantly stronger, means the secondary immune response is significantly stronger and faster, and quickly producing a large number of antibodies to neutralize the threat before it can cause illness. So this rapid heightened secondary response is the basis for vaccination as well, where a weakened antigen is introduced to generate memory cells providing long-term immunity. So you can see the memory cells, they were produced during the primary immune response. During the primary immune response, what is happening? The antibodies are being generated against these pathogens. Okay, memory cells are formed, and in the secondary immune response, because of this memory cells, there is heightened immune response. So take a look at this diagram. It visually shows the difference between the primary and the secondary immune response. Now you can see the primary immune response, the first response is slower and weaker, while the secondary immune response is much faster and stronger, thanks to the immune system memory cells. Okay, let's continue our discussion. Okay, we were talking about the primary immune response and the secondary immune response. Okay, now the primary immune response occurring during the first encounter with an antigen takes a longer time to establish immunity and involves the development of specific antigen receptors on the naive B and the T cells. Okay, so the primary immune response, it takes longer time. Here, so we can say the primary immune response, it takes a longer time to establish immunity and involves the development of specific antigen receptors on the naive B and T cells. So the initial response is slower and the protection it provides declines relatively. However, during the primary response, the immune system also generates memory cells. Okay, memory cells are generated during the primary immune response. During subsequent encounters with the same antigen, the secondary immune response kicks in, which is significantly more rapid because the antigen receptors are already present and the immune system can react more quickly. This secondary response provides stronger and longer lasting immunity, sometimes even lifelong protection against the specific pathogen. Okay, now let's talk about the B lymphocytes. Okay, so these, you can say, these B lymphocytes, they produce an army of proteins called antibodies against the pathogens. T lymphocytes, in contrast, do not produce antibodies themselves. Instead, they help B cells produce antibodies and also play other roles in coordinating the immune response. Okay, this figure we have already discussed. This is showing you the difference between primary and the secondary immune response. Okay, so let's talk about the antibodies. So these are the main molecules responsible for recognizing and neutralizing pathogens in acquired immunity. Okay, so antibodies are basically proteins produced by B lymphocytes in response to pathogens. Okay, let's talk about the structure of the antibodies, often referred to as H2L2. This means each antibody has 2 heavy chains and 2 light chains. An antibody molecule is made up of 4 peptide chains, and this is the basic building block of its structure. Now, one antibody molecule has two small light chains composed of proteins, so they are made up of proteins. It also has two heavy and longer chains composed of glycoprotein. So you can see a figure of antibody molecule here. So antibody, they are Y-shaped proteins crucial for the immune system. They are composed of two identical heavy chains and two identical light chains. And these chains are linked by disulfide bonds. Now the antigen binding sites are at the tips of the Y arms formed by a variable region of both heavy and the light chains. Okay, clear? And they are highly specific for recognizing and binding to the antigens. The light chains are shorter, while the heavy chains are longer and determine the antibody class, with their C terminal forming the Fc region at the stem of the Y, which is responsible for interacting with other immune components and triggering effector functions. So here in this image you can see there are two light chains and two heavy chains, and then you can see the variable region and the constant region as well. Okay, so antigen binding sites, they are present at the tips, you can see, and this antigen binding site, they are found by the variable regions of both heavy and light chains. Remember this. Clear? So variable region is the N terminal. Okay, clear? So there is a variable region and there is a constant region in these chains. Variable regions forms the antigen binding site. Okay, let's continue our discussion. We were talking about the antibodies. Okay, so the molecule of antibody can be represented as H2L2. Then we have seen antibody molecule has an antigen binding site. So this region is present on the antibody and this region binds to the specific antigen. Then let's talk about the disulfide bonds. Now these disulfide bonds hold the heavy and light chains together. Now type of antibody is present in the body. It is IgA, IgM, IgE, IgG and IgD. Okay, so these are also known as immunoglobulins. So is for immunoglobulins. And each antibody has distinct role in immunity. Okay, you can see this is the structure of the antibody. Okay, now let's talk about the type of acquired immune responses. Okay, now here you can see the humoral immune response and cell-mediated immune response. Humoral immune response includes antibodies. Okay, clear? Now, the acquired immune responses involves two key types. Humoral immune response, primarily mediated by antibodies, which are circulating in the blood plasma, where B lymphocytes produce antibodies that bind to and neutralize the extracellular pathogens. And the cell-mediated response, okay, or cell-mediated immunity, which is mediated by T lymphocytes, where T cells directly attack and destroy infected cells. Okay, such as cancer cells and foreign cells, providing more effective against intracellular pathogens. So you can see cell-mediated immunity, this is more effective against intracellular pathogens, and the humoral immunity, okay, or humoral immune response, this is, you can say, or this provide protection against extracellular pathogens. Okay. Now we were talking about the type of acquired immune responses. Now immune system also employs two specialized responses for enhanced protection. The memory response, mediated by long-lived memory B and the T cells, which provide a rapid and significantly enhanced response upon subsequent exposure to the same pathogen. offering immunity that can last for years or even lifetime. It also includes mucosal immunity, which is primarily mediated by IgE antibodies and various immune cells located within mucous membranes, which offers crucial first-line defense at entry points like the respiratory, digestive, and urogenital tracts against pathogens attempting to invade through these surfaces. Okay, clear? So here we talked about the memory response and the mucosal immunity. So memory response is mediated by the memory B and the T cells, and they provide rapid and enhanced response upon the subsequent exposure from the same pathogen. And then mucosal immunity. This is mediated by IgE antibodies and immune cells in the mucous membrane, and obviously this provides protection at the mucosal surfaces in the respiratory, digestive, and urogenital tract. Let's talk about the clinical significance. Okay, within this we'll be talking about the graft rejection. So in the case of organ transplant, these organs can be rejected because of the immune system or because of the immunity. Now, which type of immunity is involved in rejection of these graft or the organs? It is cell-mediated immunity. So cell-mediated immunity is the main reason why tissues and organ transplant are often rejected. So cell-mediated immunity recognizes these graft as foreign and tries to remove it. Okay. Now, to reduce the risk of rejection, doctors must carefully match the donor and recipient's tissue and blood groups. Okay, if this match is not close enough, the immune system will attack new tissues, leading to rejection. Even with the best matching, immune system can still try to reject the transplant. That's why patients need to take immunosuppressive drugs for the rest of their life. Now let's talk about the active and the passive immunity. What is the difference between active and the passive immunity? Okay, so here you can see this is a table showing you difference between active and the passive immunity. So active immunity involves the host's own body producing antibodies in response to the antigens. A process that is slow to develop but result in long-lasting effective protection with immunological memory and generating no side effects. Okay, now in contrast, passive immunity provides immediate fast-acting relief by directly administering ready-made antibodies to the host. So here ready-made antibodies are introduced in the host body, and this offers short-lived protection without immunological memory and with the potential for side effects as well. Clear? So here you can see examples of active immunity and passive immunities are also given. Example of active immunity is natural infection or vaccination. Here the body is producing the response against the pathogen, against the antigen. While in case of passive immunity, the antibodies, ready-made antibodies are introduced into the, you can say, into the host. So the colostrum, which is the mother's milk, this colostrum, it consists of an antibody IgA. So when during the breastfeeding, the child, the newborn child, it is getting these antibodies from the mother. Then through the placenta also fetus receives IgG antibody. Okay, clear? And then antitoxin injections also, like for tetanus and snake bite. This also consists of ready-made antibodies. So these antitoxins consist of these antibodies against the antigen. Clear? So these are the characteristics of active immunity and passive immunity. You can see the mechanism, okay, of active immunity and passive immunity. In case of active immunity, the host is producing antibodies against the antigen. In passive immunity, ready-made antibodies are being directly given to the host. Okay. So you can see active immunity is slow because antibodies are being produced. Passive immunity is faster. It provides immediate relief because directly antibodies are being introduced. But you can see active immunity is long lasting, but passive immunity is short lived. In case of active immunity, immunological memory cells will be there. In case of passive immunity, there will be no memory cells. Okay. Now in passive immunity, antibodies are being introduced into the body, so it may cause reactions in the body. Okay, but in the case of active immunity, there is no, you can say, no side effects because body is producing antibodies here. No foreign, you can say, particle is being introduced into the body. Let's talk about vaccination and immunization. What are the principle behind vaccination and immunization? Okay, they are based on the property of memory of the immune system. Let's talk about the vaccination process. So, the vaccine, which contains a weakened or dead pathogen, is introduced into the host body. Now, then the body will produce antibodies against this antigen. Now, at the same time, your immune system creates memory B and T cells. So these cells act like a security team that remember the invader for the future. Now, these memory cells remember the pathogen. So, if this pathogen ever tries to infect the host again, the body is ready for a quick response. Now, when needed or on subsequent exposure with the same pathogen, the body can produce large amount of antibodies very quickly. So this strong and rapid response help prevent the host or the body from getting seriously ill. Okay, so on subsequent exposure from the same pathogen, a heightened immune response is generated. Okay, so this is the mechanism of vaccination. Let's talk about passive immunization. Now, passive immunization involves giving someone preformed antibodies or antitoxins. Okay, clear? So we have already talked about passive immunity. Now, this method is especially important when rapid response is needed, such as after a tetanus infection or a snake bite. In these emergencies, waiting for the body to make its own antibodies would take too long. Now, let's talk about the modern vaccine production. Now, with recombinant DNA technology, scientists can produce the important parts of pathogens called the antigenic polypeptides. Okay, so now these vaccines consist of this antigenic polypeptide. So, antigenic polypeptide, they are the part of the pathogens against which immune response can be generated. Okay, now the scientists can produce some important parts of the pathogens which are called antigenic polypeptides. Now they can produce these antigenic polypeptides in bacteria and yeast. Okay, then they isolate this antigenic polypeptide from bacteria or yeast and they use it to make vaccines. Okay, so now vaccines can be made at large scale because of this recombinant DNA technologies, because this antigenic polypeptide, they are being produced in bacteria and yeast. Bacteria and yeast, they can multiply very fast and they can produce this antigenic polypeptide in very large quantities. Okay, example is hepatitis B vaccine, which is being produced from yeast. Clear? Now this method is safer because it is not using the whole virus, just the part of the virus that triggers the immunity. So what are these antigenic polypeptides? These are the parts of the pathogens against which immunity can be triggered. So these antigenic polypeptides are used in the vaccines now instead of the whole pathogen. Okay. Now we have already discussed this vaccination and immunization. We have also talked about the immune response, primary and the secondary immune response. Okay, let's talk about the allergy also now. So what is allergy? It's an exaggerated response of the immune system to certain antigens, which are usually harmless substances in our environment. Now, the overreaction. Okay, this overreaction is what leads to allergy symptoms. What are allergens? The substances that cause this immune response are called allergens. So, allergens can be things like dust, pollen, or even animal dander. And not everyone react to these allergens. But in allergic individuals, the immune system treats them like dangerous invaders. Okay, some common examples of allergens include mites, which are found in household dust, pollens from the flowers and trees, and animal dander from the pets. Now, the main antibody involved in allergic reaction is called IgE. Okay, now let's talk about the mechanism behind allergic reactions. Now, first, when a person is exposed to an allergen for the first time, their immune system becomes sensitized. Now, when the person is exposed to the allergen again, the IgE antibodies attach to mast cells, which are like the body's alarm system. So, this sets the stage for the allergic reaction. Now, once the mast cells are activated, they release chemicals such as histamine and serotonin. These chemicals cause the typical allergy symptoms like sneezing, itching, and swelling. And it's the body way of trying to get rid of the allergen, even though it's not actually harmful. Okay, let's talk about the allergies. What are the symptoms if a person have an allergy? So symptoms can be sneezing, okay, watery eyes, runny nose, and difficulty breathing, which should be monitored closely. Okay, now let's talk about the diagnosis of allergies. A doctor often expose patients to suspected allergens and observe these reactions. Okay, and they observe their reactions. So doctors, what they do, they expose the individual with allergens, and then they observe the reactions, what is happening to them. Okay, finally, let's talk about the treatment of the allergies. So, drug like antihistamines, adrenaline, and steroids are used to quickly reduce allergy symptoms. Antihistamine block the effects of histamine. Adrenaline can be life-saving in severe cases, and steroid help reduce inflammation. Let's talk about autoimmunity now. So, what is the definition? So, autoimmunity, okay, means the immune system is targeting its own self cells. So it's when the immune system, which is supposed to protect us from the germs or from the pathogens, it mistakenly attacks its own cells and tissues. Okay, here the immune system is not able to recognize between self and non-self cells. So scientists believe that autoimmunity can be triggered by genetic factors or environmental influences, but the exact reason is unclear. Now, result of this confusion is that the immune system damages the body's own tissues, leading to autoimmune diseases. Okay, example of autoimmune disease is rheumatoid arthritis. In this condition, the immune system attacks the joints, causing pain and swelling. Okay, now let's discuss about the immune system in the human body, which is crucial part of how we stay healthy and fight off the disease. Okay, we have talked about the type of immune. We have also talked about the type of immunity here. Okay. Now here let's talk about the primary lymphoid and secondary lymphoid organ. So the primary lymphoid organ acts as the, you can say, training academy where the body's superheroes, okay, immune cells called lymphocytes are made and get ready for the battle. Especially the bone marrow where the cells are born, and the thymus where T cells, okay, they gain their specific skills. Clear? So let's talk about the primary and the secondary lymphoid organ. Now you can see what is the main function of primary lymphoid organ: production and maturation of the lymphocytes. And then main function of secondary lymphoid organ is that they are the site for immune response initiation. Then you can see what are the primary lymphoid organs: bone marrow and thymus. And secondary lymphoid organ, these are lymph node, spleen, okay, tonsils, pairs, patches, appendix. Now these primary lymphoid organs, they are present from early development, and the secondary lymphoid organ, they develop after the birth. What is the location of primary lymphoid organs? They are protected deep within the body. And secondary lymphoid organs, they are throughout the body, okay, near potential pathogens, entry points as well. Okay, here you can see this is the location of lymphoid organs in the body. Here you can see spleen, lymph node, thymus, lymphatic vessel, lymph nodes, tonsils, and okay, now you can see here the main cell types in primary lymphoid organs. These are immature lymphocyte. In secondary lymphoid organ, these are mature lymphocytes as well as antigen presenting cells. Okay, so the primary lymphoid organ like bone marrow and thymus are where your immune system or lymphocytes are born. Okay, clear? And they learn their basic fighting skills. Clear? Now, the primary lymphoid organs, what are the key activities? The T cell development, which occurs in thymus, and B cell development, which occurs in bone marrow. And secondary lymphoid organs, what are the key activities? Antigen presentation, lymphocyte activation, and antibody production. Then, primary lymphoid organ, immune response, they are not directly involved in immune response. Secondary lymphoid organ, they are directly involved in immune response. Now, you can see the thymus, it shrinks with age. So, your immunity, you can say, decreases with the age. Okay, because thymus size is shrinking with age. Okay, but bone marrow remains active. In thymus, what happens? T cell development. So if the thymus shrinks, the T cell development will be affected. Now secondary lymphoid organs, they remain active throughout the life. Okay, let's talk about MALT, mucosal associated lymphoid tissues. So MALT is located within the lining of major tracts, including the respiratory, digestive, and urogenital tract. Okay, so MALT makes up to 50% of all the lymphoid tissues in the human body. Now, MALT acts as the first line of defense against pathogens that enter through mucosal surfaces. Now, within MALT, specialized immune cells like B lymphocytes, T lymphocytes, and plasma cells are highly concentrated to defend against the pathogens. Now, MALT can be further divided into different types based on whether it's located in the body. So, this helps us understand how the immune system customizes its defenses for different environment. Okay, so one type is GALT, gut associated lymphoid tissue, which is found in the digestive tract. So since our gut is constantly exposed to food and microorganisms, GALT plays a crucial role in protecting us from potential threats we might swallow. Then BALT is bronchus associated lymphoid tissue, which is found in the respiratory tract. So this helps protect our lungs and airways from airborne germs. Then you have NALT, which stands for nasal associated lymphoid tissue. It's a type of MALT, mucosa associated lymphoid tissue, located in the nasal passages. Okay. Now let's summarize this entire session. Now immunity is the body's ability to fight diseases, disease-causing organism via the immune system. Clear? This is what is immunity. Okay, we also talk about the innate immunity. So innate immunity is present at the birth. Okay, it includes several types of barriers: physical, physiological, cellular, and cytokine barriers. Okay, we also talked about the acquired immunity. So this type of immunity develops as the body is exposed to different pathogens throughout the life. Okay, we also read about the 5 types of antibody produced by B lymphocytes. Okay, whereas the T lymphocyte help in immune responses. Next, we also talked about the structure of the antibody. So antibodies have a special structure called H2L2. Okay. Now we also talked about the primary and the secondary immune response. So you can see primary immune response happens when your body encounters the pathogen for the first time. It's slow and not very strong. Secondary immune response is much faster and stronger because your immune system remembers the invader and is ready to fight it off quickly. Okay, we also talked about the humoral and the cell-mediated immune response. Okay, their humoral immunity involves antigens circulating in the body and body fluids, while cell-mediated immunity relies on T cells to directly attack infected or abnormal cells. Okay, now active immunity is long-lasting and self-produced. Passive immunity is short-lived and received from outside. We also talked about the allergies. So allergies are exaggerated immune response to environmental antigens, mainly involving IgE. Now autoimmunity. We also talked about the autoimmunity. So here the immune system is not able to recognize between self and non-self, and it starts attacking its own body cells. Now immune system, okay, it also includes the primary and the secondary lymphoid organ. Primary lymphoid organ includes bone marrow and thymus, and secondary lymphoid organ includes spleen, lymph nodes as well. We also talked about the MALT. This comprises 50% of lymphoid tissue and is found in major body tract linings. Let's discuss some NCERT based questions. So first question is, a patient receives a liver transplant and must take immunosuppressant for life. Which specific type of immune response are these drugs primarily targeting? So let's have a look at the answer. The immune response that will be suppressed is, okay, cell-mediated immune response. Okay, now the reason for this is that T lymphocytes or T cells are the main cells responsible for rejecting the transplanted organs. Now these cells recognize the new liver cells as foreign and try to destroy it. Immunosuppressant drug, they work by preventing these T cells from attacking the transplanted tissues. Now, without these drugs, the cell-mediated immune response would recognize the new liver as an invader and destroy it. Next question, explain the relationship between MALT, secondary lymphoid organs, and mucosal immunity. How does this system contribute to overall immune defense? Okay, so let's have a look at the answer. Okay, so here you can see mucosa associated lymphoid tissue. Okay, secondary lymphoid organs and mucosal immunity. Let's talk about them. Let's talk about the role and relationship. So, mucosa associated lymphoid tissue is a key part of immune system frontline, acting as a specialized security system within the larger network of secondary lymphoid organs, which also include the spleen and the lymph node. Now, this system is strategically located at the mucosal surfaces such as the digestive and respiratory tract to provide mucosal immunity. Essentially, MALT and the rest of the secondary lymphoid organs are where the body immune cells first encounter germs and mount a targeted defense, rather than where the cells are originally made. Now, mucosal immunity is the first line of defense at these entry points, and it uses MALT to produce specific antibodies like IgA that are released onto the mucosal surfaces where they are neutralized, where they can neutralize pathogens before they ever have a chance to enter the body and cause an infection. Okay, clear? So you should remember the role and the relationship of all of these components. Okay, so MALT, collection of lymphoid tissues along mucosal surfaces, and includes Peyer's patches, tonsils and adenoids. And then secondary lymphoid organs, these include lymph nodes, spleen and MALT. They are the sites where immune responses are initiated. And then mucosal immunity. Here you can see what is the role: immune protection at mucosal surfaces and produces Ig antibodies as well. And then role is also given here. Okay, MALT, this forms part of the secondary lymphoid organ and they are the primary site for mucosal immune response. Secondary lymphoid organ contains MALT as a specialized component and facilitate adaptive immune response, and mucosal immunity primarily mediated through MALT, first line of defense against the pathogens. Okay, what is the contribution to the immune defense? The first, MALT provides barrier protection. It means it acts as a physical and immune shield at the body's entry points. Next, MALT generates specific immune responses when a pathogen is detected. It activates lymphocytes that are specifically trained to fight that particular invader. MALT also maintains memory cells. So these are immune cells that remember past infections. Clear? Now, so if the same pathogen tries to invade again, the response is much faster and rapid. Finally, by acting quickly at mucosal surfaces, MALT helps prevent pathogens from entering the bloodstream. Let's discuss some keynotes. Immunity is divided into innate immunity, which is present by birth and it is non-specific. Okay, then there is acquired immunity, also known as the adaptive immunity, which develops over time and it is pathogen specific. B cells, they produce antibodies such as IgA, IgM, IgA, IgG. Now T cell assist in immune responses. So these cells help coordinate the immune responses that can directly attack infected cells. Now the first time your body encounters a pathogen, the response is slow and not very strong. Okay, whereas secondary response, this is faster and stronger. Now, active immunity is when your own body produces the antibodies, and this provide long-lasting protection. Passive immunity, on the other hand, is when your body receives antibodies from any other source. Vaccination is a practical way to create memory cells against the specific pathogens. So, by exposing your immune system to a harmless part of the pathogen, means antigenic polypeptides, vaccine prepare your body to respond quickly and effectively if you ever encounter the pathogen. Okay, now immune system also include primary lymphoid organ and secondary lymphoid organ. Okay, primary lymphoid organ are bone marrow and thymus, and the secondary organs are spleen, lymph nodes, MALT. Okay, MALT comprises 50% of the lymphoid tissue. Now there are some interesting questions for you. You have to compare and contrast the evolutionary advantages and disadvantages of having memory cells versus maintaining only innate immunity. Now next question is, how might understanding the H2L2 structure of antibodies help in developing targeted therapies for specific diseases? So you have to critically analyze these questions. Okay, so with this we have discussed the theory. Now let's discuss the DPP related to this chapter. Okay, let's begin with the first question. Which type of immunity is characterized by memory and specificity? So you have the options: innate immunity, acquired immunity, cytokine barriers, and physiological barrier. What is the correct answer? Here option number B is the correct answer. Okay, so acquired immunity is characterized by memory cells and it is specific, pathogen specific as well. The next question, which cells are responsible for the production of antibodies in the humoral immune response? So options are T lymphocyte, B lymphocyte, macrophages, and natural killer cells. What is the correct answer? Okay, option number B lymphocyte is the correct answer. B lymphocytes, they produce antibodies and these antibodies are present in the blood's plasma. Next question, which type of immunity is conferred to a newborn through colostrum? So colostrum is mother's milk. Okay, so breastfeeding is recommended by the doctors. Okay. Now options are active immunity, passive immunity, autoimmunity, or cell-mediated immunity. What is the correct answer? Okay, correct answer is option number B, passive immunity. So in passive immunity, ready-made antibodies are introduced into the body of another person. So here the colostrum consists of antibodies, IgA antibodies. So during breastfeeding, these antibodies are transferred to the newborn child. So this is an example of passive immunity. Next one, which of the following is an example of autoimmune disease? So options are allergy, rheumatoid arthritis, tetanus, and asthma. So in autoimmune disease, the body or the immune system is not able to recognize between self and non-self cells. So it starts targeting its own cells. So here the example is rheumatoid arthritis, option number B. Here the immune system start targeting the joint. and it causes inflammation in the joints. Next one, which lymphoid organ is the site of maturation of T lymphocytes? Option are bone marrow, spleen, thymus, and lymph nodes. What is the correct answer? Okay, option number C is the correct answer here. Thymus. So T lymphocyte, they mature in the thymus, which is a primary lymphoid organ. Okay, so with this we have discussed the DPP as well. So all our answers are correct. So with this we end this session. I hope the session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.